Thank you for the kind introduction and invitation. I really had a wonderful time ever since I came yesterday, and it's a nice hospitality to someone to come to the airport and pick you up, right? So thank you very much. And what I'm going to present to you today is some of the recent work that is going on in the lab. And those of the graduate students who are expecting to see me present a nice Western blot, you'll be disappointed. There is not much of cell line data or no Western blots I'm going to present. So almost all the work that I'm going to present to you today is from patient tissues or healthy women tissues. So that's a deviation from most of the things if you're going to hear from a cancer talk. So my lab has number of interests, and again I tell people that I'm one of the most unfocused researcher on my campus. Most of the time we are driven by the funding you get, right? But I do have some focus on certain things. So one of the things that the lab has been working on the PI3 kinase mutation, AKT isoform. We just published this last year in Cancer Research. The second work is going on is a dependence receptor pathway. It's mostly active in the neuronal system, but we see some relevance to estrogen receptor positive breast cancer. But today what I'm going to focus on is on three of these aspects: inter-individual heterogeneity in normal breast, because you heard a lot about the heterogeneity in tumors, but this is a new concept we are trying to bring into the field, and the individualized breast cancer characterization through single cell genomics that I'm going to present, and then I will move on to the genomics of organ specific metastasis. These aspects have been published, so I'm not going to cover on that. This was published last year. This has been the bread and butter issue for my lab since 1996. This is another thing we are considering is the systemic effects, and most of the people when they field think that the breast cancer is disease of the breast, but it's not true. It's a disease of the entire body. It has a systemic effects on how the muscle functions. We just had a paper which came out last month in Molecular Cancer Therapeutics. How do we use the pharmacological means to affect the breast cancer side effects using animal models? But for today, this is what I'm going to say: introduction to normal breast hierarchy, breast heterogeneity in normal breast, then characterizing adjacent normal versus the tumor of the same patient. Then I will compare the healthy breast to the tumor adjacent normal and tumor, single cell analysis, and then coming to the metastasis field. This is what we see in a normal breast. The nice layer of cells here, then it becomes DCIS, invasive, and then you get a tumor. All these years we have been focusing on the heterogeneity within this. The question my lab is interested in is, among the cell types that are here, how many different cell types exist? At a given time, the number of cells in this vary from person to person, whether that has an influence on how the tumors develop. So this is the major question we are asking: to what extent the normal breast epithelial cells are phenotypically similar between individuals. Again, most of the initial analysis at the phenotypic level, and then we'll go into the genomic level. Okay. Why are we so concerned about it? Back in 1960s, breast cancer is classified as estrogen receptor positive and estrogen receptor negative. Then we made some progress. When the microarray era came in, we could classify the breast cancer into luminal type A, luminal type B within the estrogen receptor positive, normal breast like, RB2, and basal like, and this is now replaced by what we call Claudin low subtype. And these are the era when we didn't have so-called personalized therapy. This is the survival rate, 5-year survival 90%, 50%. This used to be around 30%, no longer the case. It's more than 85% now with the Herceptin, and then we still have a major problem with the basal like breast cancer. So this is what we have in the moment. And of course, this intrinsic subtype classification has been useful in kind of predicting the outcome. Again, basal like breast cancer has worst outcome. Most of the patients who carry BRCA1 mutation have this type of breast cancer. HER2 overexpression, again the outcome has been somewhere in between. Then you have estrogen receptor positive, or luminal B and luminal A, luminal A being a much more having an overall good prognosis. And these are not just theoretical, or we can practice in the clinic. And that is one of the things that people are using is called PAM50. You can use a specific gene expression pattern, estrogen receptor, progesterone receptor, GATA3, XBP, FOXA1. they are overexpressed in luminal A type, and then you have the basal type expressing specific keratin. HER2, it's a self-explanatory, we have the HER2 overexpression. Now, the question remains, luminal type A and type B breast cancer, they will come back after 10 or 15 years in about 30% of the cases. Why is that? Did we miss a minor clone of a tumor which had a different phenotype or genotype at the initial diagnosis? In other words, most of this intrinsic classification, does it identify the major tumor clone, but failing to identify minor clone that are present at the very beginning of the diagnosis? How do we expand our ability to identify this minor clone of tumor cells in the primary tumor at the initial stage of diagnosis? So that comes the question, can we use single cell genomics to identify those minor clones? So that's the whole idea behind what we started this study. Now you may ask me a question, why are we worried about it? Here are the two cases. As per pathologist and clinician, both patients had the same subtype of breast cancer. Let us, for example, say it's a luminal type B based on the pathology. One patient, she did not develop metastasis. The other came back with the metastasis. If it is the same intrinsic subtype, given the same medication, why is that the tumors are behaving differently? We have a lot of hand-waving explanation for that. One, we can say there is a cancer stem cell hypothesis, your drug only affected the rest of them, we didn't identify the cancer stem cells, then it relapsed and came back. Other one is a clonal evolution. I mean, these are not independent, they can go together, where because of the treatment, some cells underwent different mutations, they acquired some other phenotype, it came back. Now, adding to this mix, what we wanted to ask is, it is our genetic makeup, not just the mutations that you see in the tumor, is influencing how the tumors are taking their path. In other words, how plastic are your epithelial cells, how far they can go back in their differentiation hierarchy to de-differentiate. It is wired in our genome. That's what is influencing the tumor path, in addition to the genomic aberrations that you see in the tumor. It's a mix of these two. So you may ask me the question, what is your rationale behind this thinking? There are series of papers that have come out in last few years. On the top of the list is this paper. And I always give the example of our ability to eat spicy food, right? We are all, our genome is made of the same DNA, but we still have a different ability to take our food. So that means we are wired differently. The top one said, most of the single nucleotide polymorphism which differentiates us are not located in the random region of the genome. Most of them, more than 70% of them are in the gene regulatory region, affecting how the transcription factors bind. In other words, the transcriptome that comes from the liver in my liver is different from the transcriptome in the liver of somebody else. That is because of the SNPs in that. Here is another study where they took the fibroblast, healthy fibroblasts from 62 individuals. Then they did the transcriptome analysis. Almost 50% of the genes are differentially expressed. Then you may ask the question, what is the normal gene expression pattern in a healthy fibroblast?
So, Harry, when you say differentially expressed, you mean in terms of fold expression?
Yeah, fold change.
They go in the same direction?
Not necessarily. So statistically significant difference in the gene expression pattern, right? So then these are the two papers which came out from the 1000 Genome Project last year. The level of cytokines you produce in response to a viral infection or bacterial infection varies from person to person. They have identified 17 genomic loci that determines to what extent you produce IL-6. And many of us of the same age group with the same health, if you get a flu, some of us are bedridden for a week, some of them are back to work the next day. That means our ability to respond to this infection, it all depends on the genomic loci we inherit. And this is the paper which came out last week. The gene expression affects the genetic effects of gene expression across human tissues. So there is, and again they say they have identified the interchromosomal genetic effects of 93 genes and 112 loci. That means our gene expression is dependent on the SNPs that we inherited. So considering these aspects, these are the hypotheses the lab has been pursuing. One, the level of the breast stem cell, progenitor cell, mature cell hierarchy differs between individual because of the single nucleotide polymorphism. So that changes the plasticity of epithelial cells may show individual variability. Ethnicity should be a confounding factor. Because of this, we cannot really define what is normal. We may have to define normal at an individual level rather than taking the global normal. So that's why it complicates our ability to measure the cancer stem cells. And again, the tumor heterogeneity, in my opinion, is not just derived from the mutations in the genome, but it's the complex interaction between our own genetic makeup and the mutation. That is the end product of that. Okay, the heterogeneity is coming from that. So our first task is to demonstrate the inter-individual phenotypic heterogeneity in the normal breast. Now, I brought in the ethnicity as one of the confounding factor. Why is that? We know for many years that the incidence, or at least the mortality, from triple negative breast cancer is much higher in African American women, and it's always associated with the outcome. Hispanic women have a lower incidence of breast cancer and less aggressive. In fact, the 2015 CDC report showed that overall life expectancy of Hispanic women are 4 years higher, and they have overall less cancer. Now, does the normal biology of African American women contribute to aggressive phenotype? Whenever I tried to present this, I was always hammered, because people always thought it's the socio-economic factors which are the primary determinant of the outcome. There are two reasons to not to believe that. I'm not saying it's not contributing at all. It's not two reasons not to believe that that's the only contributing factor. The one of the reason, the recent studies showed that if you compare Hispanic women and African American women with the same socioeconomic background, health access issue, still the differences persisted. Second, US Department of Defense, military, where they have the same access to health care, still the outcome in African American women was lower. And here is another example. This is the paper which came out about 2-3 months ago. Many of us, at least of my age, when we go for annual checkup, we get our hemoglobin A1c done. We don't starve like before to go and get our the fasting glucose anymore, right? But hemoglobin A1c, there is a standard. You need to have less than 5.9, or the standards are still coming down to 5.5 or 5.6 now, right? So when you do that, they go back and measure what is your circulating glucose, how your ability you're clear, because hemoglobin A1c is a glycosylation end product of the hemoglobin, which takes 3 months to accumulate. So you can't cheat by not eating sweets for 1 week. So you get an accurate measurement. What this paper said that hemoglobin A1c level to the circulating glucose level correlates very nicely with Caucasian, or people with European ancestry, but not with the African Americans. So that means we have this glycosylation enzyme activity differs based on your ethnicity. So this is another study where now people are beginning to realize that the biology of the aggressive disease, delayed diagnosis, contributes significantly compared to the other aspects, right? So this is the major focus now. And also you can go back and see the breast cancer incidence and mortality over the years. Now here is the case. This is the incidence in African American women. This is in Caucasian or European ancestry. And this is from the Native American and Hispanic ancestry right here. If you look into the mortality on the other hand, here is the thing. here is the Hispanic and American ancestry, American Indian ancestry. They are still pretty low compared to African American or European ancestry, right? So, in fact, recently there is a single nucleotide polymorphism that has been identified in Hispanic women who have more of the Native American ancestry. They have a protective allele in a gene, in the 5' region of a gene which affects its transcription factor binding. It's close to the estrogen receptor. So, there are SNPs that are not only linked to the high risk, but are also in the low risk. That's where we are. So, the first task of my lab is to demonstrate the inter-individual heterogeneity. So, what we did was to generate primary cells from healthy donors, and then the patient adjusted normal and characterize them. We characterized the expression of number of cancer stem cell marker. Then we did the genomic analysis of global and individual normal, and then we analyzed the select genes at a single cell level using on a patient basis. Now, the IU Simon Cancer Center is the only place which has a bank of normal breast healthy tissues. It is funded by Komen and Breast Cancer Research Foundation and many other organizations, where healthy women come and give their biopsies. So, we have more than 5,000 samples, and about 3 years ago my lab came up with a way where you can cryopreserve those samples so that we can pull it out at any time you want and generate the cells out of it. That is what I am going to show next. Okay? And this is what we did. We characterized these cells for CD44 and 24. These were first identified cancer stem cell markers. Then we have aldiflor, that is another type of stem cells, mostly in the proliferating cells. These cell types 44 plus 24 minus are common in basal breast cancer. This is considered more in luminal. And we also looked into CD271. CD271 are expressed in basal cells, and some of the luminal cancers I mentioned about luminal A type B, if there is a resistant clone, they may be expressing this. And this is another one which people identified as the glioblastoma, but we find them expressed to be in breast cancer. For most of my talk, focus on these one classification. 49F plus EPCAM minus are considered basal or the stem cells of the normal breast. These double positive cells are considered luminal progenitor, whereas these are considered EPCAM positive, say 49F are considered mature or differentiated cells. So, the method we used in the beginning, or this is the method which we modified to some extent. This was the first published in American Journal of Pathology, where you use the ROCK inhibitor. We have made some modification in terms of how much glucose you can use in the media and how you try to avoid DMSO in any of your reagents. So, with those modifications, we are able to do this. Again, these are the primary cells. You can now this has been gone up to more than 1,000 now. We have 1,000 cores that has been cryopreserved, and you can get the cells to pass up to at least 10 passage without any problem. You can get a couple of million cells by the time. This is how they look. Epithelial colony over feeder layer. By the end of the talk, I am going to present to you another method we came up with, because this is the mouse embryonic fibroblast. I am always concerned about having the mouse and human tissue mixed up. So, we came up with a new method where we do not even need the feeder layer, where we can grow the cells without any feeder layer of different cells. So, what we did was to harvest these cells, and this is the pattern you are going to see. CD49F here, EPCAM here in most of the cases, mature cells, luminal progenitor cells, and stem and the basal cell. Okay. Now, focus on this central part. Okay. These are the cells from 7 healthy women, and we have done many more with considering their parity, BMI, whether it's a pre-menopausal or post-menopausal, whether it was taken in a follicular phase or a luteal phase. At least with the number of samples that we analyzed, we cannot correlate to any of the phenotype. Nonetheless, what you see, this is differentiated luminal progenitor and stem cells. Here is a person, she has a lot more luminal progenitor cells, less differentiated cells, completely reverse in this case. Most of these women have only the luminal progenitor cells. These are again from the healthy normal breast. Let us look into the Aldi floor. The top is a control to give a gate, and these are Aldi floor positive cells. The number of Aldi floor positive cells varies from person to person, shown graphically. This is the distribution pattern of the Aldi floor positive cells. Again, everybody is healthy here. Now, what we did notice, however, is when we took the CD44 and 24 positive staining, these are from Caucasian. You can see the population of cells here, right? They show variability. Some of them have more CD44 positive cells. If you do the same experiment with the cells from African American women, we found this unique population of cells. They had a different profile. What we did was to take these cells and did a mini array. What we found is a network of transcription factors that went up in that unique population. There is a ZEB1, beta-catenin. They had a phenotype similar to epithelial to mesenchymal transition, in the sense these cells had, by nature, are highly plastic. They can transition between epithelial to mesenchymal state very quickly. At that time, there was a paper which came out in Nature using the mouse mammary gland, where they had identified a cell type called proCAR positive cells, which functioned as a multipotent stem cells in mouse mammary gland. When we looked into the gene expression pattern in those cells and our unique cells, there is a lot of overlap. Then we asked the question, are these proCAR positive cells also show ethnic variation in human? And that's what we did next. And that's the reference, because there are students here. If you want to see that paper, that's the reference in the bottom. It's not our paper. Here on the top is all Caucasian women. Again, this is the multipotent stem cells, proCAR positive cells, also called CD201. You see very little of those, and here you have a higher level in African American women, a little bit less in Hispanic women. And numerically, this is the numbers we see. It is highly variable. In African American, we have higher number. So then again, whenever you do these experiments, one has to be very careful, because these are not EpCAM positive. That's a measure you say they are epithelial cells. We need to make sure that they are epithelial cells. They are pan-cytokeratin positive. So there is some epithelial features in them, and they do have some of the features of the fibroblasts also. Then let us look into the Hispanic women. Now, I said we can cryopreserve these cells. If somebody who are trying to get some tissues from a different place, I can explain this story here. This was the sample which was collected in Houston. It was on a Saturday. FedEx was not ready to pull it out on that. They rushed to the airport to send me the samples in media. Sunday they didn't deliver. Monday I was chasing them throughout my campus to figure out where the delivery truck is. So what I'm trying to say is that if you are trying to collect the tissue from somewhere else, want to ship it, it still works, even though they are sitting in room temperature in media for 2 days. I was able to generate the cell lines. Okay? So if you are adventurous enough to get these tissues from somewhere, you can still get it. What do we find here is most of the Hispanic women, and again this is the differentiated cells, they have mostly the differentiated cells. At least 5 out of 7 women have highly differentiated cells, right? And various other markers show the same thing. As I told you, they have less aggressive breast cancer. Then this is the numerical value of 3 different ethnic groups: Caucasian, African American, and Hispanic. Again, Caucasian, you see a distribution like this pattern, African American and Hispanic. These are the numerical values. Now, in this day and age, we never trust what we say, right, about your ethnicity or the race. We always go back to 22andMe or somewhere to figure out what is our ancestry. That's what we did. All the 5,000 samples in the common tissue bank has been ancestry mapped with the 41 the mapping that's available in the commercially available. Here is the case. She is a mix of white and African American. It's pretty evident here because she has the pretty much African ancestry DNA here. Here she is white, but she has inherited a lot of Hispanic ancestry or Americana, because Hispanic is one of the group where I get always hammered when I go for my the grants, because how do you define Hispanic? It's a much diverse group. It can be Spanish, Latin American, Portuguese, so it becomes much more complicated. And here is 2 African American, more than 50% of the African ancestry. Majority of the Hispanic with the Americana ancestry have these differentiated cells, with the exception of this one. So there is some correlation between that. So having analyzed this, and we did find some ethnic variation in the phenotype of the cells, what we did was, then we took this approach. We took the CD49F positive, EpCAM positive cells and these differentiated cells, prepared the RNA, DNA methylation, RNA strips, and then aligned. What we did find was something unique in case of African American women. Although these cells are phenotypically differentiated, only in African American women, they had an active Wnt beta-catenin pathway, which is typically active in the stem cell component. So their transition from stem cells to differentiated cell retains some of the reminiscence of the stem cell signaling. So that way, if they get mutated, they may be able to reactivate the stem cell pathway much more quickly than the similar situation in Caucasian or Hispanic. That's the theory we have been working at the moment, but there is a lot more work to be done. Okay. So what happens to the patients who have a high risk? For example, BRCA1 mutation. Can we use this method to identify that and characterize? What was in the literature was that the BRCA1 women have more of luminal progenitor cells. These are BRCA, this is the normal. That was in the literature. But this was published using 3 patients sample in Nature Medicine in 2009, because that was so hot at that time, right? You were able to get away with 3 patients. Now try to publish 3 patient samples in the lowest of the lowest journal. You won't get it, right? When we did this in our cells, again, I'm only showing the 4 of them, we don't see any correlation between them. There is no accumulation of the luminal progenitor cells, right? What we did find, however, is that the number of cells with the aldiflor positive phenotype is much lower in BRCA1 compared to BRCA2. This becomes interesting because aldiflor positive cells are considered to be the precursors for the estrogen receptor positive mature cells. So by having lower number of these cells, the differentiation path itself stuck, so that you won't get the more estrogen receptor positive cells in BRCA mutant patients. That may be the reason why majority of the patients with the BRCA mutation get estrogen receptor negative breast cancer. So overall, to this time, my interpretation is distribution of these 3 cell types based on this marker. Again, this is a phenotypically defined cells. Doesn't mean that they are pure population. They may have 100 different varieties within them, but at least the distribution of them differs based on the ethnicity and inter-individual. We have identified the distinct population. So what determines our composition is ethnicity and genetic predisposition. That determines the heterogeneity in our normal breast. When we are doing all this work, there was a paper which came out in JCO, Journal of Clinical Oncology. What they found, what African American women have a higher incidence of basal and mesenchymal stem-like triple negative breast cancer, and they have much higher intra-tumor heterogeneity. That means they can shift between them. The cell type that I explained to you is normal cell type, which is enriched in African American women, have the same mesenchymal stem-like phenotype. It is possible that the cell type origin of tumors in African Americans may be different because they have much higher number of these mesenchymal stem cells. They may be subjected to the mutation. So there may be an original difference between that. So with all these resources, there are a number of opportunities we get. One, now we have immortalized cells from these women based on the ancestry map. You can start manipulating them, right? So you can resource the genomics and epigenomic and transformational studies. You can do all sorts of genomics and cytop. Some of you may know that we can further classify these cells. And the question we can ask, scientific questions, does the ratio between the stem, luminal, and mature cells at a given time, as we age, can we use that as a parameter to classify, to determine the susceptibility to cancer? This is something my lab is interested in. And what we did here is the case. For example, this is from a Hispanic woman here. Before immortalizing, her cells were all differentiated. As soon as you immortalize, they become luminal progenitor. Majority of the breast cancers originate from luminal progenitor cells. So the very first step in the transformation process may involve the cells acquiring this phenotype, right? We don't know at the moment. There are technical glitches here. We don't know. It could be that only these cells are getting immortalized. That's why we have it. Or immortalization itself has this de-differentiation capacity, which we don't know. We tried single cell immortalization. Those experiments didn't work.
Sorry, but in that experiment, you actually immortalized them by genetic modification.
Yeah,
right?
H-TERT overexpression. H-TERT overexpression.
Do you have any case where spontaneously?
No,
like
we have never.
no.
There was one case of BRCA1 mutant patient. She behaved like that for a couple of passage, then it went away, right? And this is the immortalization in case of Caucasian and African American. Again, they are prior to that they were here the luminal progenitor. Immortalization, we do get more of the stem-like cells in this case, but not in this case, right? And we have created a number of this. Again, we have ancestry mapped all of them. The top are Caucasian. You can see the lot of European ancestry here. These are Hispanic. You can see a mixture of all these. She's a African American. She has more than 50% of the African DNA in her. Now, one of the questions that has been around for a number of years has been, what is the meaning of intrinsic subtype of breast cancer? Does it mean there is a normal counterpart to that in the normal breast from which they originated? These cells served as a model system for us to examine that. What I'm showing here is the PAM50 that is clinically used to classify the breast cancer into different subtypes using this 50 gene signature. When we subjected our immortalized cell lines to that purpose, we were able to classify our cells into luminal A, basal, and normal. The breast cancer field suffered for many years with the non-availability of the normal cells which represented the luminal characteristics. Almost all of us have used MCF10A, which is a basal. HMAC cells that we use, it's basal. But we have been able to generate these cells for the first time like this, right? And here is the other one. When we did an unsupervised clustering of our cells, these are our cells, and the MCF10A and HMAC, you can see our cells are completely different. With the exception of the two cell lines we created from African American women, for those unique cells that I described, CD201 positive EPCAN, they cluster with this MCF10A and HMAC. Majority of our cells have highest expression of luminal gene. Because of the interest of the time, I'm not showing the data where we can show they express estrogen receptor, FOXA1, GATA3, which are representative of the luminal mature cells. It's the first time we have been able to generate those cells. I just wrote the first draft of the paper. Once it is published, these cell lines will be made available to anybody who needs it. But it's not an easy cells to grow, okay? And then we use the keratin staining pattern to further classify. For example, keratin 14, which identify the basal cells, our MCF10A control cells. It's not, keratin 19 is not expressed. MCF7 is a luminal subtype cancer cells. Keratin 19 is not. Our cells are either double positive in this case or completely basal. In another case, we have more towards the basal, and some of them are luminal cells. Then we ask the question, there is inter-individual variation in the differentiation. Do these cells differ in inter-individually? The way we did this experiment is we grew these cells in mammosphere. It's a kind of an assay which will allow you to self-renew the cells with the stem cell activity. They can also differentiate. The question we asked is, do these cells differ from person to person, the ability to form these mammospheres as well as differentiate? Now, here is an example. What do you see? Again, the cells in this compartment are called differentiated cells. These are luminal progenitor cells. These are stem cells. Caucasian, Caucasian, Hispanic. You see the number of cells here compared to her, compared to her, compared to her. You see the differentiation capacity from the healthy cell lines that we created varies from person to person, right? This is not unique. Then next we did was what we call the mammosphere assay. This is the 2D culture, the way the cells look. In mammospheres, the shape and the size of the mammospheres, sorry, this is not mammosphere, matrigel, acini formation varies from person to person. That is also linked to the differentiation capacity and ability to interact with the extracellular matrix. At the same time point, if you do this assay, you see a personal variation. Now, there is, I know you are in an engineering school, there is always an interest in developing organoid cultures, the in vitro culture which we can mimic the cancer situation. What we did, we came up with the two assays we are trying to do in this. The top one is the hydrogel assay. You can see our cells forming a duct similar to the normal breast. If you do the keratin 14 staining is most of the cells are on the outer side, and this is the keratin 19. Some of them are outside because they are double positive, and there are few keratin 19 cells which are internal. So, we are able to generate these cells now into the duct similar to the cancer, the normal breast. The idea is now you can hit them with the different oncogenes or knock down certain tumor suppressor genes to see whether you change this ductal morphology. This is in a collagen gel. You can see the cells now forming again a duct. What we see the difference here is here mostly the double positive cells forms this ductal structure. So, the type of cells that can form this ductal structure depends on the collagen matrix or the matrix that you use. Hydrogel has collagen, laminin, couple of other extracellular matrix protein, but this is purely collagen, right? So, this is all the work we did with the normal. Now, one of the major interpretation we can make at this stage is normal varies from person to person, both phenotypically and differentiation. That begs the question, if you are characterizing the tumor, what should be your normal control, right? That is the next step we did. What we did was to culture the tumor and normal from the same person and then analyze them. Look in the top, this is a differentiated tumor from a patient. Her adjacent normal, she is mostly luminal progenitor cells, and here is her tumor, differentiated population as well as luminal progenitor. That is perfectly fine. Here are the two cases with the moderately differentiated tumors. Particularly pay attention to this one. This is from a Hispanic woman. Her adjacent normal is mostly differentiated cells. Her tumor is luminal progenitor. Now, the question is, those who are doing the gene expression analysis, if you take her own normal cells and compare to her tumor, you are going to get thousands of genes being differentially expressed. How many of those differential expression is because of mutations or genomic aberration in tumor compared to the two cells are being in two different differentiation status? If I take these cells and these cells from the normal breast, for example, I see more than 2,500 genes being differentially expressed. So, that will be carried over here. So, the identifying the genes that are differentially expressed in tumor because of genomic aberration is not as simple as we always envisioned before. You need to take into account this differentiation cascade. And the thing that I am going to tell you in the next one is, here is the case particularly for graduate students, if your mentor comes and says, or the reviewer says that why are you not focusing on the highly differentially expressed gene, think back and twice and say that I don't think that is functionally important. That may be something linked to the differentiation. Okay, so you need to do all sorts of analysis before you pick up your gene. And here is a case where same patient had in one breast one type of breast cancer, in another breast she had a different type of cancer. You can see they are different, phenotypically different. In the same genetic background, the cancer itself changes the pathway of differentiation. Okay. So, with all this data now, I think I may have confused you even further, because you can't even use her own normal cells because it's in a differentiated state. What do you do then? How do you start coming down to further refining your analysis? So, the next thing we did was we took the CD49F positive, EpCAM positive cells from healthy donors, tumor adjacent normal, and tumor from the same patient. So, we kind of reduced the, again narrowed the gap. When we did that analysis, what we found was there are 26 genes which are highly overexpressed in adjacent normal compared to the healthy. That means the adjacent normal itself has undergone some changes because of the field effect or the distant effect of the tumor. And these 26 genes form a network of an inflammation. That means the normal breast itself has an influence of the tumor because of the inflammatory climate in that.
Is normal from the same breast or they're different?
Now, that's an issue. I could only do it from the same breast. I could only do it from women who underwent mastectomy. We took them as far as away from possible. From the clinical point of view, maybe the contralateral, but is it practical to ask women when they are going to a surgery to open the one breast to say, can I get a piece from the next breast? It may not be practical, but that may be the ideal normal to use for comparison, right? Assuming that the field effect hasn't gone to the other breast, but that's a different story, right? So, the interpretation I always said, the majority of the gene expression difference between normal and tumors can be attributed to differentiation status. That's the caveat which is affecting our interpretation of signaling network. How many of you have used the Ingenuity pathway or any of this, all this David? We always use it, right? But the pathways that you see may not be really truly active in cancer. It could be because the cells are in two different status. So, then you pick up this pathway, buy all sorts of drugs available against your pathway and put it in the culture, nothing happens. You are scratching your head where the things went wrong. It could be that's the reason, right? So, highly differentially expressed may not be functionally significant. Of course, the essays that I presented has its own caveat because I did not consider the tumor microenvironment. Still, it raises the question which tissue should be as a control. So, then what we thought, okay, we still have the problem with our analysis. What if we do the analysis at a single cell level? Can we identify the heterogeneity, right? This is the C1 Biomax system that we have. We are slowly going to go into the 10x genomic study. What we did was, we took the normal cells and tumor adjacent normal of the same patient, gated for the aldiflor positive cells, then we went for 49F and EpCAM positive cells, and these population we took about 50 cells from each and did a qRT-PCR for about 96 genes. This is the PCA plot, tumor and normal separates very nicely, and when you do the unsupervised hierarchical clustering, the tumors are separated nicely, the normal here. The main thing which comes out is when you do a violin plot. Here, this is the distribution pattern of this gene in each cell of the tumor. This is the normal. Wnt5a, you can easily say the tumor is overexpressed. The question comes, look into this FOXA1 here, right? If you did a normal genome analysis, you would say the FOXA1 expression is lost in the tumor because the level is low. But in normal, you see two different patterns, some cells expressing very high level, some expressing at the level similar to the tumor. An alternative interpretation is that the tumor originated from the cells which naturally did not express FOXA1. So, I wouldn't target that gene as a therapeutic target in this case, right. So, those are the kind of assays you can do. Having done this, we thought, okay, we can further extend this. because this came from the paper in Science where they did was they took the glioblastoma. When they did the whole genome analysis, the glioblastoma was classified into one subtype. When they did the single cell genomics, they were able to identify 4 different subtypes of glioblastoma in the same patient in tumor. They had different treatments for each of them, but since they did in the bulk, those treatments are missed. So, we thought let us do the same thing to identify minor clones. Again, I do not have to introduce to you the PAM50, which we have done before. This is the one we did. What we did was we took either there are normal counterpart to this PAM50, are these genes really overexpressed or not, and can we identify the multiple subtypes in the same tumor. So, we did 2 different ways. We took the normal and tumor of the same patient, took the epithelial cells bulk, that is one analysis. From the same patient, we took the phenotypically defined population, so that you get rid of the effects of the differentiation. How does it compare? In the PCA plot, what you see is a complete mess when you do the bulk. If these defined population, which is supposed to represent the most undifferentiated population in the normal breast, they kind of make a nice pattern. This is what happens if you do the bulk analysis. The red are the normal cells, they are scattered everywhere. There is a one cluster of tumor cells, there is another small cluster of tumor cells, these are the green cells, right. This is the bulk analysis. If you do the same thing with the phenotypically defined population, you see the majority of the normal clustering here. Right next to is one subtype of breast cancer, which is enriched for the basal enriched genes. Then you have a luminal, then luminal basal combination. This is from the same tumor. You are able to identify 3 subtypes of tumor. This is another patient. If you do the bulk analysis, you identify only one major clone of tumor. If you do the phenotypically defined cells, you identify a major clone and a minor clone. Let us get back to the original question we had, which gene is really differentially expressed? Again, with the phenotypically population, you find a different expression. The same genes show a different pattern of expression when you have unselected gene. So, this brings the question, which gene is really overexpressed or underexpressed in tumor? It all depends on what you use for comparison. So, my cynical view, precision oncology, is it an illusion or are we there yet? We need to ask the question. How can we achieve this particular pattern of success? A yoga practicing baby to a 76 year married life still going strongly. Based on the data I presented to you, you can look it up like this. We can take this path or feel pessimistic, glass is half full or half empty, whichever way you want to take it. Or we can start thinking differently. We are all different. So, we can take a path that we have to start thinking about clinical trials on an individual basis. We need to understand the individual biology, their genetic makeup, then decide what works for the person. Is it practical? I am not sure. I am not a clinician, but it is something we can start thinking about as we move forward. So, having done this, there are additional advantages to the method we presented. So, one of the things, how many of you, I am sure many of you are doing the genomics. For you to identify a mutation in tumor with the current technology, that has to be present in at least 10 to 15% of the tumor cells. Otherwise, you won't detect the mutation that easily, right? No matter how deep you go. So, one of the things we thought is, now we have this culturing system. I am not saying it is without any caveats. Every method we use, every model we use has its own drawbacks. But with this method, can we let the minor clones of tumor cells to expand, at the same time get rid of all the stromal cell contamination in your DNA preparation, and increase our ability to see the minor clones with additional mutations? So, the way we did was, we took the germline DNA, took the tumor, mush-mashed it, made the DNA from one portion, The other portion we cultured it, grew the cells out, purified them, and did the same sequencing. What you see is that the unprocessed tumor and tumor cells, these are the common mutations you see, including the one which is the most famous one. But in the cultured cells, we were able to identify additional mutations. This is not the artifact of culturing, because we had the normal cells cultured at the same time as a control. They didn't have those mutations. These are not hotspot DNA which gets mutated when you put into culture. So these are unique to the tumor cells. So suggesting that our method allowed us to expand them. We used droplet digital PCR to verify and to make sure that the new mutation, particularly FOXO1 mutation, we could see in the tumor cells. Then we thought of asking, can we expand this to metastasis? Now, it is becoming apparent that the metastasis is no longer the same as the primary tumor. They evolve independently. So we are trying to see whether the metastasis we can grow, because most of the treatment decisions are made based on the characteristic of the primary tumor. But metastasis evolves independently. The latest data says that even before the tumor is diagnosed, metastatic cells have already left the primary tumor. They are already homed in a different organ, evolving independently. So we need to change the concept of doing the drug discovery, not based on the primary tumor. We need to think about how we can get the drug discovery based on the genomic characteristics of the metastasis. So with that in mind, here is a liver biopsy of a triple negative breast cancer patient. You can use this method to grow very clean. That metastasis has all the luminal progenitor cell properties. And then when we did the whole genomic sequence, it's a triple negative breast cancer. Primary tumor should have multiple genomic aberration. This one is pretty clean. She has only one translocation. What we did find, however, she has mutation in many of the genes that hits the epigenome. So you don't have to have tons of mutation for this metastasis. Few mutation which affect the epigenome, we were able to see that. And we have extended this to other cancers. This is choroidal melanoma that has metastasized to liver. As expected, it's a mess, right? You can see so many chromosomal translocation. This is the spindle cell carcinoma. We extended this to multiple other cancers, including the colon cancer, the pancreatic cancer, brain tumor. But what we did find is that irrespective of the cancers that from which one got the liver metastasis, all of them had the mutation in the pathway that is linked to TNF and TGF-beta. These are the mutations we found in different types of liver metastasis from different tumors, but they all link to one. So there is a liver nest phenotype in this. So we further developed a new method which I mentioned to you before. This is from the normal breast. We can develop this method. We can identify multiple cell types in the normal breast, including grow out the basal and myoepithelial cells. We can grow out the cells which are considered luminal progenitor. We can also grow out the cells which are called EPS cells, endogenous pluripotent stem cells within the normal breast. Very rare cells, these are called EPS. We can grow them out. With this method, we can grow out the mesenchymal stem cells from the normal breast. And these are the 4 cell types we can grow: the EPS, luminal, myoepithelial, and mesenchymal stem cells from a single core biopsy from a healthy breast. And this method is useful to grow even the normal breast as well as tumor. You can grow the pleural effusion from a breast cancer patient, which goes very nicely here. This is again the pleural effusion itself is very heterogeneous with the number of subpopulation of cells you can see. You can also grow the pancreatic neuroendocrine tumor growing in liver. Again, you can identify the heterogeneity. You can also use these cells for the drug discovery to identify drug response. For example, this is a ovarian cancer, the ascites fluid from which we can grow the cells. If you treat them with the cisplatin, only certain population of cells, in this case here, these EpCAM positive cells are the only one which are targeted by cisplatin, but these populations are resistant to it. The same thing you can do with the different markers. So the cells we can do is the cells we can get from this method can be used for the drug screening purpose. So this is the overall conclusions I have from my presentation. The normal breast shows the remarkable heterogeneity among individuals. Ethnicity and genetic predisposition is one of the factors. Again, we need to take into consideration differences in the differentiation between normal and tumor before you judge whether the gene is specifically overexpressed or underexpressed in a tumor. And we may need to start using purified population of normal and tumor from the same patient to identify some of the cancer specific gene expression differences. And we can use this technique to grow the metastasis and single cell genomics. So, at the moment, the breast cancer incidence and outcome is multifactorial. You have our own inter-individual heterogeneity in the normal breast. We have ethnicity dependent differences. Then we have these other factors, the impact of obesity on normal and cancer stem cells, and socioeconomic factors. Only these two are the factors which is in our control. Other one we have inherited, nothing we can do about it. So, this circle is the one which will determine the outcome from breast cancer. Now, I presented this in showing you a happy face all the time. Do you think I had a happy face when I was trying to publish this? Absolutely not. I had many days I felt like this, and many days I felt like this when I tried to publish, all right? Because this is a new concept, people don't want to believe that there can be heterogeneity in the normal breast. So, although we had a couple of papers, finally I published the initial observation in this one, and these are the number of people who have been working in the lab of the past and the present. They have been extremely helpful in getting the thing, and without money we are nobody. So, these are the funding agencies which helped, including Susan G. Komen, Vera Bradley Foundation, VA, and recently we got a grant from the Falk Medical Trust just about 2 weeks ago. This is the current lab. So, with that I will stop here and take any questions you have.
Questions from the audience, please?
Yeah.
Thanks, it's an impressive study. I have several questions, but I'm just going to start with one. Do you have a longitudinal study from a patient that gives that phenotype that is normal for your cells and eventually develop
Yes.
cancer?
We have the samples from more than 20 patients who developed breast cancer after 5 years. Natasha Marino in our group, she is somewhere here in the Komen. She has been working on that. What she has done was to laser capture micro dissect the normal breast tissue from these women, because we don't have the breast tumor tissue because these women went to different oncologists, we can't get it. But she has taken those normal breast tissue from them, laser capture micro dissected, and then about 60 other age matched normal who are still healthy. So, she has done some genomic work, but I don't think she has published it yet, and it's in the preliminary. With respect to normal breast, I believe we did get the IRB approval to do the longitudinal study, even the normal, so that we can control for the age associated changes. Those things are going on. Yes. Yeah. Okay. Yeah.
So this is a very interesting study. So my questions are coming more from a mechanical or engineering framework. So you have addressed the heterogeneity, but we don't still understand what is the origin of that heterogeneity.
So
more and more studies that are showing that the environment plays a positive or a negative.
Yes.
So my question is, when you take these cells and then when you culture them on the similar environment, which could be a flat substrate,
right,
are we masking those genes that we are trying to isolate and look for, which have been hidden off all this while, which could be the key?
That I agree, because once you take out the things, it does mess up the environment. So, there is an ongoing study in the lab. What we have done is to ask the question in a slightly different way. What we have done was we have laser capture micro dissected epithelial cells from Caucasian and African American women, 12 each. So, they are in the same in vivo environment, right? Those are being sequenced now. We should be able to address some of those ethnicity dependent differences which is not influenced by my culturing method or put it on this different environment. So then we can go back and forth to come up with the genes which may be
Then a follow-up to that would be, do you think that the origin of heterogeneity has plasticity in it? In other words, like as the
Yes, yes,
tumor is growing, for example.
yes,
So instead of looking at what the differential genes are,
yeah.
can we also take a back step and try and look at what the environmental contributions could be? Is there a way that we can look at that?
See, let me put it this, the single nucleotide polymorphism I've been emphasizing on, that activity of those polymorphism may be influenced by the environment, right? But it will be incredibly hard to model this in an experimental setting, right?
From evolutionary point of view,
Yes.
let's say that you have a tissue has grown evolutionary to have all the same type of cells,
Yes,
right? So, but then there is heterogeneity.
yes,
With increasing phenotypic differences,
yeah,
do we see more apoptotic events that could lead to?
it is possible. For example, I told you the Hispanic women with the Indian ancestry, American Indian ancestry having a protection. We had 50 cryopreserved samples from Hispanic women. 2 of them had that gene. When we took that out, those cells, prepared the cells from them, the one with those ancestry had low stem cell activity, higher apoptotic activity. So some of the single nucleotide polymorphisms we carry confers different phenotypic characteristic to these cells. How much that is influenced by the environment, not even the internal body environment, extraneous environment, it is something, it takes years and years of work, I believe. Yeah, yeah,
I think we have time for one more question.
yeah, yeah.
I think the key is to use the sisters study, right? I mean, it's just, identical twins.
Yes, yes,
So
yeah, yeah. That's a
That's the key.
yes, exactly. That is, I don't know whether they will be able to get those samples. So that will be really, yeah.
Because-
An identical twin study will be the best one to do it. Yes, where we can dissect out the environment versus your genetic makeup, how to what extent it differs. Yes, I completely agree. But it's not everybody is after sisters. For every study, they need them, right? So, a much sought after group. Yeah.
Okay, let's thank our speaker and as the last question of the presentation.
Thank you. Appreciate it. Thank you.
The speaker will be here for another 5 minutes if you would like to ask him a question.
The guys that sign up for lunch, can you please wait
My name
here?
is Amrinder Nain. I'm from mechanical engineering. So I regulate fibrous environments in a very precise way to control the plasticity of cells. So we can have induced in 3D, 2D, and stuff.
Okay, that's wonderful.
Is it okay if I can send you email?